In this video, we're going to take a brief historical look at the development of some of our understanding about key components and features of the immune system. The content covered in this video will be discussed in much more detail in other videos, but for context, it is a good idea to understand some of the incredible discoveries and scientific research that has led to our current understanding of immunology. Historically, people recognised some of the properties of immunity even before knowledge of pathogenic microbes or the presence of the immune system existed. One of the earliest recorded acknowledgements of immunity and the immune system came from the Greek historian Thucydides, who wrote about a plague that swept through Athens in 430 BC, noting that people who had recovered from a previous bout of the disease were able to nurse the sick and the dying without suffering from the illness a second time. Yet it was with those that had recovered from the disease that the sick and dying found most comfort. These knew what it was from experience, and had now no fear for themselves, for the same man was never attacked twice, never at least fatally. Indirectly, he was referencing the ability of the immune system to produce immune memory, upon which vaccination is based, and the ability of the immune system to learn and remember how best to defend against a disease. Although Edward Jenner is often credited with the development of the first vaccine in 1796, using cowpox to immunise against the deadly smallpox virus, the principles of deliberate exposure to a pathogen to try and prevent a further infection have been used across the world for a long time previously, even as far back as 1000 AD in China, where people learned that inhaling the powdered crusts of smallpox scabs, a procedure called variolation, could help protect recipients against future bouts of the disease. What Jenner does need to be credited for is realising that he could use the safer cowpox virus to immunise against smallpox, which was far safer and easier to manage than using the smallpox virus itself. Jenner's work eventually led to the outlawing of variolation. In the 18th century, the French scientist Pierre-Louis Moreau de Maupertuis observed that some dogs and mice were resistant to the effect of scorpion venom, which was another early recognition of the effects of acquired immunity. The following century, Louis Pasteur and Robert Koch built on these observations further, with Pasteur conducting work on vaccination and proposing his germ theory of disease, whilst Koch's proofs in 1891 confirmed that microorganisms were responsible for infectious diseases. Viruses were described as human pathogens 10 years later by Walter Reed, who discovered the yellow fever virus. Immunology made huge strides forward and took off as a discipline in the 19th century with the discovery that the immune system comprised both cellular and soluble components, or what were called humoral components at the time. The Russian zoologist Ilya Mechnikov is credited with the discovery of phagocytes, which are cells that are able to engulf, consume, and remove other things, such as cells or foreign debris. In his seminal experiment, Mechnikov inserted a rose thorn into a transparent starfish larvae, and observed that within just a few hours, that cells had surrounded the foreign object and proposed correctly that these same cells would respond in the same way to an infectious pathogen, which was the basis of his theory of cellular immunity. About the same time, Shiabasaburo Kitasato and Emil von Behring proposed the humoral theory of immunity after observing that the serum from a rabbit that had been infected with diphtheria was able to protect a rabbit that had not been previously exposed to the pathogen. Without knowing it, they were describing the actions of antibodies and a phenomenon called passive immunity. The work of Paul Ehrlich expanded on these findings, proposing his side chain theory, which helped expand the understanding of humoral immunity and the specificity of antibodies. More recently, it has become understood that both the cellular and humoral theories are correct, and the focus has shifted to understanding how and why the immune system becomes activated. Frank Macfarlane Burnett proposed a self-non-self theory, stating that only non-self entities activate the immune system, but this theory was incomplete. Technically, food is a foreign entity. It certainly isn't part of the body, but it doesn't trigger an immune response. Therefore, the immune system must be more intelligent than just seeing self and non-self. To expand on this theory, Charles Janeway proposed the infectious non-self theory, stating that only entities that express conserved pathogen-associated molecular patterns activate the immune system. But if this theory is correct, then why doesn't the immune system destroy the millions of friendly bacteria that line almost every surface of our body? They express the same molecular patterns as pathogens that cause disease. Perhaps the best model we have at the moment, which is by no means perfect, is the danger model, which was proposed by Polymatzia, which says that the immune response is triggered by danger signals from damaged tissues. One weakness of this model lies within organ transplantation, which is beneficial to the recipient, yet often causes an immune response. So where does that leave us? Well, immunology is incredibly complex, and there is still a huge amount that we do not yet understand. The immune system continues to surprise and amaze us, and these theories will continue to be refined as we uncover more features of this remarkable protective system.